I'm Sourya, an instructor in La Jolla Institute for Immunology. And my research topic is epigenomics, particularly to understand the 3D chromatin structure, gene regulation, genetic variants, and their functional implications. And here I have come with a novel concept called interaction QTL or IQTL, which is basically SNPs associated with chromatin looping. So as you know, the conventional EQTL studies like this SNP, which is shown to be associated with this RMDL3 expression in a genotype dependent manner. And this was found in one of our lab's earlier study back in 2016 in Nature Communications. At the same study, we found that the same SNP also shows allele-specific change in chromatin interactions. Like for the risk allele, the interaction is between RMDL3 and the ZBPP2 gene. Whereas for non-risk allele, the target gene switches from ZBPP2 to IKZF3. Okay, so we wondered whether we can have a framework where we can get this SNPs which are showing genotype dependent trend of chromatin interactions. And here we found that the same SNP in exactly same loci, we found a chromatin interactions where this genotype dependent trend of chromatin contact mimics that of gene expression. Okay, so we term them as IQTLs, these SNPs, that interaction QTLs or QTLs showing change of chromatin interactions. So to derive the IQTLs, here we used high-chip data. High-chip because it shows much higher precision regulatory interactome compared to other protocols like Hi-C. And here we chose acetylation as the protein of interest because acetylation is the active enhancer mark, and we chose naive CD4 as the target cell type. So using these 30 donors high-chip data, we got first of all high-chip loops using one of our earlier work, fit high-chip, which identifies the statistically significant high-chip loops. So using this fit high-chip loops for 30 donors and simultaneously using the genotype information for all those 30 donors from the DICE database, which is our in-house database of EQTLs. You can check this website for the details, and which was published in Cell 2018. So using this genotype information and high-chip loops, we modeled these two statistics. One is the genotype dependent statistic showing the change of chromatin contact between different genotypes, and also the allele specific statistic, the change of high-chip reads between two allele. So both of these statistics are fed in a QTL discovery framework known as Rascal, which was originally developed for 1D QTL discovery like ChIP-seq or ATAC-seq, but here we adapted it for chromatin interactions. So Rascal has three different models. Default model, in addition, it has genotype dependent allele specific models. So we applied all of them and finally combined them and filtered them. In addition, we also did the pair t-test of allele specific reads, and we combined all of them. The final set where we got around 9,000 SNPs, IQTLs, and associated 2,000 high-chip loops. To post-process or validate, we got transcription factor binding motif analysis, overlap with regulatory ChIP-seq or ATAC-seq peaks, overlap with reference EQTLs or HQTLs, overlap with GWAS SNPs, and finally also open target genetics and association function knowledge portal was also used to validate these SNPs. So first of all, we found that IQTLs, their effect size is concordant with respect to the reference blueprint histone QTLs and also reference DICE naive CD4 EQTLs, showing high concordance and statistical significance. But we also found that 36% IQTLs were not EQTLs or HQTLs, and 28% were not even in LD with them, suggesting that there are IQTLs independent of those reference QTLs. In addition, we found that 57% IQTLs are also EQTLs in naive CD4 T cells, 12% are not EQTLs in naive CD4 but EQTLs in other CD4 T cells like Th1, Th2, Treg memory, etc. And 31% IQTLs are not EQTLs in any CD4 T cells. So we wanted to characterize these three sets of IQTLs, and we found that first of all, IQTLs which are common to naive CD4 EQTLs like 57%, so they are showing high degree of disease heritability as computed by stratified LD score regression. Actually, we adapted that to compute the heritability enrichment for a given set of target SNPs like these IQTLs which are common to naive CD4 EQTLs, and we found that IQTLs or even that common ones, these two sets are showing higher degree of disease heritability compared to the standalone DICE CD4 naive EQTL sets. In fact, the IQTLs which are exclusive to any CD4 T cell EQTLs, these are also showing high degree of disease heritability. So it suggests that by IQTLs we are getting important disease informative SNPs. In addition, we found that the same set DICE, that IQTLs and also IQTLs common to naive CD4 EQTLs, these are showing high enrichment of fine mapped GWAS SNPs compared to the standalone DICE CD4 naive EQTLs. and these are for all different such immune diseases and fine mapped Jiva SNPs, suggesting that by this IQTS we are getting important SNPs, either fine mapped Jiva SNPs or high disease heritability conforming SNPs. The second thing that we found, that IQTLs which are not naive CD4 EQTLs but in other CD4 T cell EQTLs, like this MICB gene. This is a very important gene for T cell response, and we found that this MICB gene is differentially upregulated in memory Treg cell type. So we found that these two SNPs which are IQTLs and which are not EQTLs in CD4 naive T cell but EQTLs in memory Treg. In fact, this 321, this SNP is actually fine mapped EQTL for memory Treg, whereas this SNP, this is a EQTL and also showing CTCF binding, suggesting or showing this region of chromatin interactions in this locus. So it means that the change of chromatin interactions in this locus is getting translated to the change of gene expression in the derived CD4 T cell types. The third one that we found, that IQTS may not always be associated with one or two loops. Like here, this IQTL, this was associated with two loops, out of which I have shown one loop as an example, this genotype dependent and allele specific trend. But we also found that same IQTL can also alter chromatin connectivity in a broad region. Like this RNA set2 gene, this is a very important gene for chromosomal rearrangement. We found that change of genotype dependent chromatin connectivity, as you can see by this color scale, see by all of these 78 chromatin loops, this is showing similar directional change of genotype dependent change of chromatin contact count. And also this is the aggregated heat map of chromatin connectivity, and this score on top of them, this showing the average connectivity, and you can see the steady increase of chromatin connectivity across different genotypes towards the alternate homozygous genotype. So we term this SNP as connectivity QTL, which are affecting chromatin contacts in a broad region. And overall 80% of this connectivity QTLs we found are also EQTLs in naive CD4, which is understandable because usually the change in chromatin conformation in a bigger scale is also associated with the change in gene expression. So by IQTS we could come up with a set of SNPs which are showing significant change in chromatin contact, also associated with the gene expression, or also carrying higher disease heritability or fine mapped Jiva SNPs. So you can find the GitHub repository and WashU browser for a lot of examples. And with that, I want to thank my supervisors, Dr. Farhat Ai, and all other LGI and our LGI lab members for their support. And looking forward to interacting with you all and discussing about these projects and other projects in the conference. Thank you so much.